तीन 3 महिन्याचा रिजेक्ट टाकलाय.
हेलो தோசை தர போட்டோ வரல. facing. ऐसे तो
Ma'am, good evening, ma'am.
Good evening, Nirmala.
Ma'am, good evening, ma'am.
Good evening, ma.
Welcome, ma'am.
Thank you.
Good evening, ma'am.
Good evening, ma'am.
Display wallpaper, live wallpaper.
Haribalan sir.
Yes,
Welcome to
yes,
our
ma'am.
program. Welcome. Sir, good evening, sir.
Uh, uh, Good evening, ma'am. Just give me a few minutes. I will.
Okay, sir. Okay. Ma'am, shall we start our program, ma'am? Ma'am? கேட்கவே
A few more minutes, Nirmala.
Yes, ma'am. Yes, ma'am. Thank you, ma'am.
ஐயா ஓடாதம்மா, வழுக்கி விழுந்துருவோம்மா. ஏமா அப்படி ஓடுற? செருப்பு கேட்டு விடுமா, செருப்ப பார்.
ரெடி ஆயிக்கோ.
பல்வாய் போடும். ஓடா, தின்னு தின்னு ஓடாத. தண்ணி வலிக்கிடும், புரியுதா? நடந்து போய் நடந்து வா, சொல்லிட்டேன்.
Nirmala?
Ma'am, start பண்ணுவாங்க.
Yeah.
Yes, ma'am. Thank you, ma'am. Good evening to one and all present here. Revered Director Samba, Revered Secretary Amba, esteemed Madam Principal, Honorable Chief Guest, faculty members from our institution and other institutions and students. Good evening to one and all present here. It gives me an immense pleasure to deliver a welcome address for this one-day international webinar on research perspectives in life sciences, organized by the Department of Zoology. I extend a cordial welcome to our beloved Ambas, esteemed Madam Principal. Our Madam Principal supports us in many ways, not only giving suggestions, but also with the execution of those ideas into action with a smiling face. I'm equally happy to welcome our Controller of Examinations. I offer my heartfelt welcome to our resource person, Dr. Haribalan Perumal Swami, Research Professor, Department of Agricultural and Biotechnology, College of Agricultural and Life Sciences, Seoul National University, Korea. I am happy to welcome our next session resource person, Dr. K. Kadiraman, Associate Professor of Biotechnology, University of Madras, Chennai. He will come and shortly join with us. I would like to express my gratitude to our honorable resource persons who have accepted our invitation immediately without any hesitation and gracing the webinar with their presence. Nowadays, science clearly evolves along with the advances in knowledge and technology with the societal concerns. Both the scientific community and the broader society, which is embedded and become increasingly concerned about safety, environmental protection, treatment of animals and human. Life sciences highlight the flexibility of the process of science to accommodate new concern and build upon new opportunities. I ensure this webinar will take towards the new panorama in life sciences. Thank you. May I invite our Madam Principal to deliver the presidential address.
Respected resource person, beloved head of the Department of Zoology, dear faculty members and participants of this webinar, I wish you all a very pleasant evening. During this lockdown period, we have been organizing plenty of webinars, FDPs, quiz programs to engage and enrich the knowledge of the staff and students. This webinar is one such event organized by our zoology department. My hearty congratulations to the head of the department and organizers of this webinar for the effort and initiative taken towards this. In the address, I would like to give a small intro and insight into the life science. Life science is an enormous scientific field which answers to some of the basic fundamental questions about ourselves. As the name suggests, it is the study of life in all its form, past, present, and future. It explores each and every area of the planet where we live in, like land, oceans, deserts, mountains, and forests. It also includes the study of plants, animals, viruses, bacteria, single-celled organisms, and even cells. It is the study of the huge blue whale which reaches upon the ocean for air, to the tiny small ant which crawls in our kitchen corner for sweet, and to the bacteria which helps us in our digestive process. The study of life science look into how we live, where we live, and how we might have a better living. The study also aims to learn everything about life on our planet. The study of life science lends an important insight into the disease processes. It helps to develop some novel therapies and then to invent modern, innovative medical equipment and thereby directly improving human health. The life science can be combined with other branches also, like engineering and sciences. With engineering, it could be bioengineering, biomaterials, and biomedical engineering. And with other sciences, for example, with mathematics, it could be biomaths. With computer science, it could be biocomputers or bioinformatics. And with physics, it could be biophysics. And with anthropology, it could be bioanthropology. Am I audible? Hello?
Yes, ma'am. Yes, ma'am.
Am I audible?
Yes,
Yes,
ma'am. Yes, ma'am.
ma'am. Audible, ma'am.
With anthropology, it could be bioanthropology. With astronomy, it is astrobiology. And even with linguistics, it could be studied as biolinguistics. So, life science has a wide reach. And there are around 8.7 million animal species and 4 lakhs plant species and plus bacteria and viruses roam the world. Each one has its own form of life. So, the branches of life science is plenty, and to list a few: ecology, botany, zoology, entomology, microbiology, cell biology, physiology, genetics, epidemiology, paleontology, and marine biology. Ecology, as a branch of life science, is the study of each organism with its interaction with the environment. It also includes the food chains and the parasitic and its behavior. That is how the species are related to one another and with the environment. That is about ecology. And botany, we know it's the study of the plant kingdom, right from herbs, shrubs, creepers, and it includes trees, algae, fungi, everything in the plant kingdom. Zoology, we know it is the study of the animal kingdom. It studies about the behavioral pattern, breeding pattern, migration, and the habitat of all the animals in the animal kingdom. And entomology is the study of creepers and crawlers like insects, worms, and snails. Microbiology is the study of the smallest of all living beings, that is the viruses and bacteria. That is about microbiology. And then cell biology, as the name itself says, it is study of cells. We all know that our body is made up of lot of cells, and each cell has its own tiny ecosystem. So the cell biology is the study of that tiny cells. And then physiology. Other branches of life sciences consider a living being as exists in the surrounding environment. But physiology considers how the organism stay alive, what makes it to be alive. And so it deals with the organ system, organs, molecules, and cells, and also with the chemical interaction between them so that it makes the person alive and his behavior. And then genetics. Genetics is the study of genes, and it traces back to the Mendel's period and is study about the bees plant. And the genetic study about the traits which passes down and how it adapts itself to the environment. And next comes the epidemiology, which is the study of epidemics like this COVID-19. What causes that epidemics? What is the reason? What virus or bacteria causes that? How it breeds, how it lives, how it affects the person, how the virus dies. So that is about the epidemiology. And next branch is the paleontology, which is the study of the organisms which are not alive today. For example, dinosaurs. So it is the study of how those organisms might have lived with the fossil record we have and the remains, the preserved remains of those organisms. And the last branch is marine biology. And as the name suggests, it is the study of all the living organisms that exist in the ocean, right from the huge whale to the tiny fish, to the plankton to the algae. And these are all some of the branches of life sciences, though there are many. I listed out only a few. So, while studying the life sciences, it gives us a vast knowledge and understanding about the planet where we live in, so that we could have a greater appreciation for the planet that is our house, and then it gives us the importance to keep it and to protect it. So, I once again congratulate the Department of Zoology for having chosen an apt topic, life science, for this webinar, and I wish the webinar every success. Thank you, and over to the organizers.
Thank you, ma'am.
Thank you, ma'am. Thank you, ma'am, for your elaborate classification of life sciences, ma'am. Thank you, ma'am. Now I invite Dr. B. Kalaivani, Assistant Professor of Zoology, to introduce our first session resource person.
Good evening to one and all. First resource person, Dr. Haribalan Perumal Sami. He is now works as an research professor, Bioactive Natural Products, Department of Agricultural Biotechnology, College of Agriculture and Life Sciences, Seoul University, Seoul. He was completed his BSc in Nehru Memorial College, Puthanambatti, Trichy. He did MSc in Bharathiar University and also did PG Diploma in Microbial Biotechnology in the same university. He did PhD in Agricultural Biotechnology in Seoul University, South Korea. He did PDF in Microbiology and Natural Product Chemistry from 2012 to 15. Then he attained research professor position in Cancer Biology, Microbiology and Natural Chemistry from 2015 to till date. Apart from this, he obtained 50,000 USD from National Research Foundation, that is NRF Korea, from 2019 to 22. He is actively participated in the YouTube channel Korea Tamil Nanbargal Kulu. In that, he took classes for Korean language. He also helping the candidates who is going to South Korea. From this brief introduction, now I invite Dr. Haribalan.
Thank you very much for your introduction, and also happy to be part of this international webinar conducted by the Sarada College for Women. I'm very happy, and also we have very less time, so I'm directly going to present because we have consecutive one more seminar, so I don't want to be elaborate more detailing. And once again, thank you very much, everyone, for your organizing such a great webinar. These days in India, uh, this is the way we can execute our scientific research and everything. Even though this pandemic situation, we are challenging through this kind of internet platform to share our research and everything to everyone. It will give us broad vision. We can contact anyone from in the world. It's like a very huge platform to communicate each other. So I'm very happy, and yeah, I hope it will be interesting. And since I have very less time, because I'm keep on taking webinar, usually I take more than 2 hours also sometimes based upon the elaborate session or some questions and everything. But yeah, actually they were saying that I don't have time to, so they reduced it 1 hour. So actually, that's why they reduced it 1 hour. Otherwise, I would have extended more. It is okay. If you have any doubt regarding this, you can get contact from Professor Kalaigorani or some other professors. If still I'm ready to have all your questions and everything if we have time. Otherwise, you can send me email or we can collaborate near future also. So I don't have any issue. So directly I will go to my topic. Just give me a few minutes. Yeah. Can you see my screen?
Yes.
Can you see my screen?
Yes, sir.
Sir.
Okay, thank you. Actually, I have prepared so many number of slides actually, so I will explain one by one, very deep, not very detailed. Even though we all are from biotechnology, zoology, and biochemistry, all background. Before going to start, I must say that I am also a zoology student. I did my zoology in BSc zoology, and now I'm in Korea, and I'm taking research in cancer part. So why I'm saying this line means we already know, right? Those who are studying zoology and everybody thinking and everybody saying that you are geology, you are taking class in geology, or you are taking course BSc geology, and our friends and many people were that thinking and view onto us something different, right? So I'm the one of the example for you all. So don't need to be getting some complex regarding this, because yesterday we were talking a small session with Professor Kalevani and Professor Umarani, that those who are having geology background, uh, they are surviving. They have the basic knowledge. They have the all type of biological knowledge compared to studying only for biotechnology, biochemistry, something like that. Just the name, like a branding name, having their they thought something different. But I must say, I would say that geology is the one of the important thing, in particularly in the UG courses. You should have geology subject, and all our staff faculty members are more excellent, and also they have more enormous experience to teach, how to teach, and we will have basic knowledge also. And still I can talk any topic in biology background because of my professors from undergraduate from Nehru Memorial College, Economics. So they have been teaching a lot to me when I study in undergraduate. That still it's helping me to interact all the subject in biology or biotechnology, biochemistry, and everything actually. So just I want to share this information to all our people. So be confident. You can achieve more than me. You can achieve wherever you are from geology background. So just want to convey this message to all of you. Anyway, so today I'm going to explain about what we are doing in our lab, and at the same time just to give the brief explanation about biosynthesis of nanoparticle, and then how green synthesis technology we are following for the cancer treatment. So that's what we are going to talk about today. Biomedical approaches mean there are so many biomedical approaches like antibacterial, cancer, anti-inflammatory, and so many things. So all are interlinked or interconnected. I will give brief introduction and then I will move on. And everybody knows that cancer is a global killer, global disease, and we usually frequently these days we are getting this kind of diseases, and from our relative friends, all our known community, we are always hearing this word, right? The cancer, cancer. Why we are getting the cancer? Why we don't get cancer in before actually? Because these days there are so many food habitat and our lifestyle and everything has changed, and also the external factors like contamination, food contamination, air pollution, and so many other things have been influenced us to have this kind of disease actually. And I keep on saying that everybody have a cancerous cell. It's not a disease actually. Everybody have a cancerous cell. This is one kind of cell. This cell, when the cell undergoes some process, which I will explain later. Before that, I will just explain that cancer is not a disease. It's like a caused by bacteria or virus or something like that. It's like a cell improper of cell function or dysfunction of the cell. Then the cell become cancerous growth or tumorous growth. Then only we are getting this kind of disease actually. So it's not by any particular microbes
ஹலோ
or any particular
சார்,
virus actually.
நான்
That's
போய்
why,
நான் இன்னும் செக் பண்ணல. இப்போ என்னன்னா
and
ஹோஸ்ட் அவர்
also that,
கொடுத்துருவாங்க.
these days we are getting these kind of disease because of our, like I said, external factors, right? Some of the external factors like all food contaminated, food addictive, and pollution, everything is influencing our immune system dysfunctional actually. If our immune system is not functioning properly, then the cells become cancerous. How they become cancerous, I will explain briefly. And before that, you can see the purple color in this graph. But in Asia, 48.2% of incidence of the cancer, and same 57.3 people were dying This is the 2018 global map they are showing. Why the Asian people death rate or incidents are very high? Because we don't have proper health checkup and we don't have awareness about what is the cancer and actually when we have to detect or how it will be cured. We don't have any knowledge regarding this, particularly among the public people rather than the research community actually. Because we do have regular health checkup once in a year actually. Western countries or highly developed countries, they will have a health checkup regularly once in a year. Then only they can detect the cancer in the earliest. Because if you detect cancerous or tumorous growth in initial stage, it will be curable. Of course, 100% curable. If you cannot detect in the first stage or second stage, it will be very, very difficult to cure actually. So this what the Asian population of incidence and death rate is very, very high because our people, we always go to hospital once the symptoms shown actually. Cancer is something different. It's not a disease like I said. It's not like you will get fever or you will get some pain at the earliest or at the beginning stage. In the beginning stage, you don't get any kind of symptoms unless you will do regular health checkup. If you don't do regular health checkup, you definitely you will have some kind of these things. We cannot detect at the earliest actually. But this is the biggest threat. There is the biggest problem in Asian community actually. From all our Asian countries, we are always don't give importance to our health actually. That is the big problem. And being women community, I would say that breast cancer, you know better than me, you don't have any kind of symptoms or any kind of tumor by your naked eye or something because it's a lobule, right? It is a lobule or soft tissue. Unless proper health checkup, you cannot or you cannot find that exactly whether do you have the cancerous tumor or not because it's like a lobule or soft tissue. It's a hide inside the tissue actually. That is the biggest problem being the woman population in the worldwide. So if you reach age around 40 or 35 to 40, you must have regular health checkup once in a year to avoid this kind of problem actually. So please do have health checkup once you are in age 40 around. After 40, you should have 2 years once or once in a year. You should have the regular health checkup. Then only you will be, because of these days, like I said, so many external factor which influences immune system. Once the immune system not functioning properly, then you will get the cancer actually. Okay. We everybody know that cancerous cells is like not like normal cell. It's like if they can divide continuously, they don't have proper regular shape and size. And yeah, this is the image which we already know, right? The normal cell system how it divide, and whereas in compared to the cancer cell, you can see that image. It will be they cannot control. They don't have one signal to control in our body. Once we will get the cancer cell. The very, very important thing I'm going to say about now, it's called apoptosis. So we can see, sorry, the apoptosis. The apoptosis is a programmed cell death. I will, for example, if one cell, they have the lifespan for 2 weeks or 3 weeks. Once they reach the third week or second week, their cells should be removed or destroyed from our body actually. If the cell skip this process, then the cell become cancerous. That means the apoptotic process is already like a computer programming. They already fixed because when the new cells are coming from bone marrow, they will have a lifespan. Every cell we have the lifespan in our body. Once it removed from our body, then new cells can be synthesized. This is the biological process every time. If the cell skip this process, then the cell become cancerous. Then they cannot control or they cannot kill by our immune system or immune body. Like I said, why our immune system is mainly involved means our immune system have T helper cells and other immune cells very active. If something unknown process or if something
Haribalan,
yes ma'am.
please mute Dr. Parimala.
You can mute.
You are the host, no? You can mute. Dr. Parimala. Next, yours.
Okay, thank you.
Okay. If any noise, you can mute.
Okay.
Okay.
Yes. Like I said, the apoptosis is the very important process in our body. If the cell skip this apoptotic process, then the cell become cancerous actually. So this is the main thing we should know as a biologist. We should know we are the biologists, right? We should know how the cancer is forming. You should explain to the public people and give awareness to have a regular health checkup, and you should have the how the cancer is forming, the main sources, something like that. So that's why I'm explaining. So apoptotic process is a very important process. If the cells skip this process, then the cell become cancerous. If normal apoptosis means the cells will be destroyed or removed from our body once the lifespan is done, and then new cell will be formed from bone marrow. So this is the process called apoptosis actually. You can see the right side, you can see the blue color cell. This is like a cancerous cell. These cells, it's something is mutated or something dysfunctional of the immune system. You can have this kind of enormous cell growth in your body. And also, now I'm going to explain about very important thing. We always have some confusion, right? What is the benign tumor? What is the cancerous tumor? We do have confusion, right? If I have some tumor in my body, I always confusion and unnecessarily worrying and something like that, right? So, being a biologist, we should know what is benign tumor, what is the tumorous or cancerous tumor. Benign tumor always present in the same place for long period. They don't grow and they don't give any symptom, and they don't invasive. Invasive means they don't move one place to another place. We can call it as invasive or metastasis means uh, they can move one place to another place. They can spread one tissue to another tissue. This process is called invasion or metastasis. This is the process. So they don't undergo, and they will be stay in the same place for prolonged time. You no need to worry about the study. Whereas in the cancerous growth cell, they can be invasive, they can be metastasized to one place to another place, one tissue to another tissue, and then they can be easily spread actually. So now you can ask how they spread actually. This is the very important thing. You should know that when the cancer cell want to move one place to another place, they will produce their own blood vessels actually. So this forming of own blood vessels by cancer cell, this process is called angiogenesis. I will explain in next slide. The angiogenesis is the very important process. For example, if we give biopsy, or if we give the biopsy, or we give some CT scan image, the doctor always check this is the cancerous or this is the benign tumor. How they check? Please remember now. If you see cell mass or cell growth in the image, and also you should check whether in around the surface area, some square centimeter, if you could see some blood vessels in around the cell mass, then it's definitely cancerous cell. If you don't see any blood vessels in around particular centimeter of the cell mass, it's not a cancerous cell. This is the real detection. This is the final
ஹலோ,
confirmation.
நான்
All the doctor will
அவர்கிட்ட
do
சொல்லி மியூட்
actually.
பண்ண சொல்லிட்டேன். இப்போ சார் 5 மணிக்கு
So
தான் வரேன்னாரு. அவர் வர்றப்ப ஹரி தான் ஹோஸ்ட், அதனால அவன் பார்க்கறானா என்னங்கிறது தெரியல. முடிச்ச உடனே நம்ம சாரை இது பண்ணிக்கலாம். அப்படி இன்கேஸ் வந்தாருன்னா சொல்லிடுங்க, கொஞ்சம் இன்ஃபார்ம் ஆயிடுங்க. முடிச்ச உடனே நம்ம அவரை இன்வைட் பண்ணிக்கலாம், அக்செப்ட் பண்ணிக்கலாம். உடனே நம்ம ஹோஸ்ட் மாறின உடனே. ஆமா.
So,
அதனால என்ன
this is the main reason actually. We have to detect. So when we go for the doctor, always they will check
அப்படி இப்ப
that.
அகில மாத்தி விடுறது பிரச்சனை இல்லை. மாத்தி விட்டா இவனுக்கு மாறுவான். ஷேர் பண்ண முடியுமா?
Excuse me.
அதுதான் பார்க்க வேண்டியதா இருக்கு. அதுக்கு தான் பார்க்க வேண்டியதா இருக்கு. பரவாயில்ல, போயிட்டு இருக்கட்டும், பார்க்கலாம். முடிஞ்ச உடனே நம்ம இது ஷேர் பண்ணி.
Okay. So this is the main step of angiogenesis process. Doctors always check there is the blood vessels in around the cell mass. If there is the blood vessel, it's like cancerous cell growth. If you don't have any blood vessels in around the cell mass, it's not a cancer, it's just a benign. So you have to confirm like that. If your friends or somebody approach you, you should explain this is the main mechanism. How they detect it, simple malignant or simple benign tumor. And second step, they will go to biopsy and then they will check remaining process. Okay, anyway, so hallmark of cancer, like I said, is like cell sufficiency growth signal. They can divide continuously. No one can control by externally our body. They can control by themselves, so they can divide continuously, and they avoiding apoptotic process. If the cell end of apoptosis, the cell will die, right? That's why they skip apoptotic process. And sustained angiogenesis means they can be develop their own blood vessels. We can wonder, right, why they need blood vessels? Because if they want to move one place to another place or a nearby tissue, but how about, for example, if I have cancer in lung and if I have cancer in tissue, I mean liver also, I mean how they can transfer one place to another place? Because all these finest vessel forming and enter into the main bloodstream, right? Then main bloodstream carry the blood, carry that cancer cell, and then they can move, they can spread in around our whole body. Actually, this is the process. The cancerous cell want to grow, so they are then only. That's why they are making their own blood vessel through angiogenesis process. This is the main characteristic of the cancer actually. So the same thing, benign and malignant. I given some images for you. You can see the normal benign tumor, the size and shape also always same, right? Whereas you can see in the down figure in the green color image, there are three different stages. The final stage is called invasive cancer. Actually, you can see so much burst, so many things is burst. You can image some one circle, right? If the circle is filled, then what will happen? It will be overlapping, and finally it will be burst, right? Then it will be burst, and then the burst cell can enter into the small blood vessels, and then small blood vessels enter into the main bloodstream. Then only they can move to nearby tissue. This is the process usually they do it. This is the final stage of the cancer. It's called invasive or metastasis process. Okay, with the angiogenesis, you can see the initial stage. You can see the cell mass in around, right? They can make they produce small blood vessel initially, and then they can grow. Once the stage will be first or second or third stage, that will become very severe, right? This is the final stage. You can see the figure E, tumor vascular, the figure. Uh, that one is so many branches has formed, right? It would help to spread nearby tissue and attack more other organs. This is the process called angiogenesis, very important process in cancer biology. And same time, invasive and metastasis, like I said, is the fourth stage, is the final stage of the cancer actually. Yeah, this was the brief introduction. How the cancer is. Like I said, cancer is not a disease. It's not a disease. Cancerous, it's a cell. Everybody we have. Even now, everybody we have that kind of cell. But our immune system working properly, so that apoptotic process or other process is continuously working on, right? Then one cell become old, they completely removed or engulf or something. So many biological mechanisms like autogenesis, engulfing, phagocytosis, and apoptosis. So many processes. They are helping to remove completely old cells or damaged cells or unknown cells, completely removed every day. So this all, then only we are getting very fresh, because When we are fresh means a new cell forming, continuously formation from bone marrow. This is the biological functioning programming keep on running and keep on working in our body. Once this immune system failure because of our food habit, because of some external factors, it is affecting our body, then only that cell getting mutated, some immune system or something getting mutated, or dysfunction of the immune system or dysfunction of the WBC cell like a T cell, H cell, or so many things, then only the metastatic process, then they become cancerous cell, and then they become very vigorous and they can divide continuously and making tumor. This is the simplest mechanism in our body we are having actually. And there are so many cancer treatment options nowadays available. I will explain briefly. And immunotherapy is kind of antibody treatment. For example, I will inject antibody, or I will inject some antigen into our body. Then automatically it producing antibody. That antibody itself can be improve our immune system. It's not directly killing or directly affecting the cancerous cell. It's indirect method actually. Then radiation therapy, we everybody know that, right? The radiation therapy only applicable at the initial stage. That's why I'm checking if you are detected, if I'm detected cancer as the earliest stage, it's 100% curable through radiation therapy. And also we already knew that so many side effects. Once the person undergo the radiation therapy, if the person get good immune system, he or she can be recovered from this process. Otherwise, the side effect itself cause very big problem. And target therapy now we are looking at, and so many researchers, scientists, we are looking in target therapy. That's why we are making nanoparticle, so many things which I will explain detailedly. And chemotherapy is a chemically synthesized drug. Nowadays, drugs also become getting resistance, and they are not active against to the target specific. And also, once we making chemically synthesized drug, there are three clinical phase in available. If one drug become marketing, they should first undergo stage 1, stage 2, and stage 3. Each stage have 10 years actually. That's why some drug will be passed phase 2, some drug will be failed the phase 2, or some drug will be failed at phase 3 because of the chemically synthesized have some side effect, or and also it will be affect the non-target cell actually. And bone marrow transplantation is like expanding our life. For example, already being the cell become affected by cancer means our body need new cells, right, for functioning the normal function. Yes, yes. So if bone marrow transplantation, it will be really helpful to produce more cell. Then only the normal function will be done same time, even though you have the cancer. But it will be only extending the life. It's not a permanent cure. And surgery also, like I said, only possible at the earliest stage, on first stage 1 or stage 2 only. And hormone therapy, it's like hormone imbalance. For example, if you have the breast cancer, even a man also have some kind of breast cancer because of the hormone imbalance. And you also, particularly the woman population also, if hormone imbalance, it also indirectly having breast cancer, right? If we changing into the hormone balance exactly, it's like a prevention method rather than once we have the cancer already, and it will be very very difficult to control if you are in third stage or fourth stage. It's also possible indirect method to have to cure the earliest or initial stage, or prevention from the getting cancer. And these are the things I want to explain that from 17th century itself, the diseases already have, but the name may be different. These days we are calling it as a tumor, and earliest they may call it as a different name. But in the time, it's not that much like frequenting detect from the human population. But these days we are getting this kind of disease because of our weak immune system, or our functioning of our food, and everything is indirectly affecting the immune system. Then all the biological process has failure, and then we are getting this kind of diseases. And you can see the red color around the circle, right? There is no new naturally available drug is approved from this region. In this topic, we are focusing on natural product from plant basis, and why we want to plant-based drug actually. So which is very very important, because our ancestor we are having all food as a medicine, right? That's why they don't get any kind of disease. But these days we have been changed a lot to the Western habitat. We are eating so many Western style food, burger, pizza, fried chicken, and so many things, and also so many food additives also we are having, right? These itself having some side effect and indirectly affecting the immune system, then we are getting this kind of problem. So now we have to go back, and we have to looking for what kind of a plant or medicinal plant or food they have been taking in order to avoid this kind of disease at the earliest. And also one of the reason to have this kind of food as a medicine means it will be very very easy to getting past the clinical trial in different phases, because if the drug synthesized in chemically, it will be the toxic and it will be failure to pass this process actually. If you have food as a medicine, it will be definitely it will be passed to the all the phases, and it will be have good drug, and also very cheap, and because of the abundance of the enormous amount we have, and particularly India have biggest resource in medicinal plants actually. So if we explore these kind of plants, and if we do research in these things, then it will be really good for us to have good medicine for the cancer therapy. So that's why we are looking on this kind of medicinally available food as a medicine, natural product drug. And like I said, the immune system, yeah, I will give just brief. There are two type you can image. One is M1 phase or M2 phase. M1 phase always be immediate response, and M2 phase is always be the secondary response, something like that. So this is the very very important to maintain the balance actually. If that pro-inflammatory or anti-inflammatory should be maintained. If pro-inflammatory goes very high, this itself to kill the normal cell actually, we will getting some kind of side effects. So this is some kind of thing. Since we don't have time, so I will skip this one. Just remember, if your immune system is very good, then you will be very strong, and you don't get any kind of disease actually. And it is also related to the immune related system. And nanoparticle, I will give the nanoparticle nothing but just a small sized. The size is 100 nanometer to 1 nanometer, and they can be produced using so many metal like gold, silver, zinc, polymer, and so many things. And the shape may be round or cube or rod, and anything. And also the surface we can be attached using that functional group like acid or amine or so many things we can attach. Then we always believe that nanoparticle only for physics people or chemistry people, but we do have synthesis nanoparticle as a biologist or biotechnologist is very easy. And also we are using bacteria or yeast or fungi to have confirmed this whole type of nanoparticle. And we wonder, right, how bacteria synthesis nanoparticle, right? So I will explain. These are the metal we usually use for nanoparticle purpose, because these have the less toxic to the normal cell actually, gold, palladium, selenium, and everything. And before that, I just will explain the mechanism how bacteria is synthesizing nanoparticle. Just imagine bacteria cell wall, right, outside or inside. Bacteria have so many enzymes. So if our, for example, when the gold metal, when I dissolve into the water, it will give positive and negative charged ion actually. It becomes charged actually. So just 1 minute, there is some noise. So this is the very very important mechanism. I will explain. We are making a bacterial with the help of bacteria, and bacteria itself to producing or synthesizing the nanoparticle. The mechanism is when the metal dissolve in the water or some other medium, it will become solid, right? When it becomes solid, it will be charged like a plus or minus charge, right? When we put the solution into the bacteria surface have attractant of plus or minus charge. So they can be easily attract into the cell wall, and then this metal and then our compound like a plant material or compound we mix together with gold, right? And then we add together, right? Then this metal coated our compound or metal coated plant extract when enter into the bacteria when attached into the surface of the bacteria, that yields itself having attaching and attracting, and then that become the chargeless form. For example, Au plus become Au0, or some, if for example if silver means Ag plus become Au0. Au0 means reduced form, that is means removal of charge and become more small sized aggregated form. This is called nanoparticle synthesis mechanism. For example, a big sized compound becomes small sized compound, a nano sized compound. The process, bacteria have some enzyme, that enzyme making nanoparticle through this kind of mechanism actually. Just remember like that, okay? So once the bacteria enzyme is very helpful, so they can synthesize our nanoparticle through the help of all our extracellular intercellular. You can see this slide, intercellular synthesis or extracellular synthesis, right? When we add all these aqueous solution, like I said, this is the gold nanoparticle. When we put into the water, it becomes charged, right? When they enter into the inside the cell, the bacteria enzyme, the enzyme making this Au charged ion into reduced form. Reduced form means more small form or removal of the charge. When the removal of charge, it become more small sized and then looks like aggregated. This metal aggregated is called nanoparticle. This is the mechanism. If that aggregation formed inside the cell of bacteria is called intercellular synthesis. If aggregation formed outside the cell is called extracellular synthesis. This is the mechanism. You may wonder, right, how we know this is exactly a nanoparticle or not, right? This is the mechanism, and same time we have to do in the practical aspects, right? So we usually do have some kind of ginger family, like we Korea called as a ginseng, the same like gingerous family. They have huge, enormous amount of biomedical application, and then we have isolated some compound, and also the bacteria which we are using is a probiotic bacteria. It's a beneficial bacteria, it's not a harmful bacteria, okay? This beneficial bacteria, for example, it is helpful to make milk into curd, or some other purpose of bread making, so many things. And also we have intestinal beneficial bacteria, right? It will help to for absorption, for digestion, it is helping. So these kind of bacteria we are working on it, and this bacteria have some kind of enzyme. That enzyme itself, it helping to convert a big macromolecule into micromolecule with the help of some metal or some gold or silver, zinc, so many metal ions. This is the mechanism actually. And you can see this, we are culturing bacteria, and then we add all the chemical into it and waiting for 12 to 24 hours. Later you can see the purple color is aggregation is forming, right? This is by, we can detect through by eye formation of the nanoparticle. And I'm saying this is the nanoparticle, right? But nobody will believe, right? So we do have characterization and confirm. And also I have been explained the intercellular and extracellular, so many thing by theoretically, right? I do have I do have a result to prove my theoretical explanation, right? So for that, anyway, this is the examples of nanoparticle we have been doing. Okay, so first detection after the purple color coming, we do have the UV. So based on the UV, we will do only bacteria, only compound, only cold, all mixed together making nanoparticle. First step, you can see the formation of nanoparticle, and the second figure is the XRD, the gold accumulation. You can see the 111 is the gold accumulation peak, and also FTIR is the functional group attachment compared with the normal before or after making nanoparticle. And particle distribution. Particle distribution means that whether it is small, uniformly small sized particle or not. If it's a big sized, then it's not a nano, right? So we have to do the particle distribution. And stability. Stability means if I do one time, nanoparticle is formed. If I do second time, if don't form, it's not good, right? So I have to be like a temperature dependent, pH dependent, concentration dependent of gold, concentration dependent of plant or compound. So many things we do have stability or optimization to confirm. So many time to confirm that whether we are getting same result or not. After that, we do the very important thing is the TEM analysis. You can see the bacteria and outer surface. There are so many black colored dot, right? This is nanoparticle. Why is outer surface of the outside only there is a black color spot, right? Not inside the black, inside the bacteria. This is called, we are saying, the extracellular bacteria, because this type of bacteria and plant material is attached only in the surface of the bacteria and produced surface itself. It's called extracellular synthesis. Whereas you can see in the down figure, I mean figure D, so you can see the down figure, there are so many accumulation only inside the bacteria. It's called intercellular bacteria. So this why we are saying that is extracellular synthesis bacteria is a, sorry, extracellular synthesis of nanoparticle, and some bacteria have synthesis intercellular actually. So this is the first confirmation. We do have biological TEM analysis to confirm exactly. And then there are so many other images for the nanoparticle accumulation formation. For example, if I say that this is the gold nanoparticle, I do have evidence, right? So that's why we do the element analysis for accumulation of highest number of gold. If I have the silver nanoparticle, if I have the zinc nanoparticle, I should have all element analysis and so many other analyses to confirm exactly. This is another type of bacteria. It's a flower shaped, and also we do have the zinc nanoparticle is a different shape and size. So all the thing we should have confirmation exactly, and whatever we have given theoretical explanation, we do have the practical aspects to prove completely all the result and everything. Then only we should exactly conclude that evidence with based upon our result and everything. And like I said, this so many cancer cell we have been culturing in our lab, and we do have treatment. After once we confirm all the nanoparticles, first we do the MTT analysis to have check whether it has activity or not, and then we also do have the normal cell confirmation also, because most of the drug will be failure if they have side effect to the normal cell. So if food as a medicine, if we choose natural product, then only we can be avoid this kind of problem once our drug passes into the phase 1, phase 2, and phase 3 for human or clinical trial actually. So that's why every time we should have compared with the normal cell line and then cancer cell line. And this image, you can see the right side, left side, you can see the control cancer cell after treatment completely degraded, right? Whereas in this is the breast cancer and this is the stomach cancer, because I have been frequently doing this one, so I know exactly by the size and shape I can detect. So you can see the control and treatment, dose treatment and manner completely destroyed with our nanoparticle treatment. And also this is a very important confirmation. Why we do synthesis nanoparticle, why we don't use ascites, because all plant material have secondary metabolite or single compound, right? Why don't we use directly as a single compound? Why I should do convert it into the nano? This is a very important thing. Once I converted into the nano, that compound become nano sized, right? That will be really very helpful to enter inside the cell very easily and accumulate or target specific. Like I said, target specific treatment is very important. Because if I treat some compound, it's a macromolecule secondary metabolite compound, that compound can attach outside the surface, and then they couldn't enter inside because of the size is very big, right? But if I convert into the nano, they can easily enter inside the cell. They can become the target specific, even very less size and even very less amount will be very, very good. So this why we also detect in the higher magnification cyto-vivo analysis. You can see the black color in the figure C. There are so many nanoparticle accumulation. In the page figure, you can see the green color accumulation is targeting inside the cell organelle, whether it can be target nucleus, mitochondria, lysosome, or so many other cell organelles. It will be most target specific and it will be very, very easy. That's why we are converting macromolecule or secondary metabolite into nanoparticle for more specific and target treatment. This way. And also these are other nanoparticle images. You can see the white color, so many accumulation inside the cell. And also we do have the biological term to confirm, see where exactly, this is the nucleus, where exactly, you can see the black color spot in here and here, different type of cell. You can see the accumulation compared to the control. After 3 to 4 hours later, it can be entered easily, and then they can become target specific and kill the cancer cell. So this is what we confirm, and also we do confirm with fluorescence and effect and everything. Okay, I just will be showing because we don't have enough time. So and also we have the ROS isolation staining, because ROS we already know that free radical, right? If uh, if normal cell have free radical, that free radical should be removed from our body, otherwise it will be killed by that free radical itself kill normal cell actually. But whereas in cancer cell, we should produce more ROS, right, by the treatment of our compound. So then only it can be killed in around the cell actually. So this is why we also check ROS staining. And also, like I said before, like apoptotic pathway, so many other pathway, molecular pathway, autogenesis, and engulfing phagocytosis, and so many molecular pathways available these days. So we do have confirmed, then only we should exactly confirm that this nanoparticle involving this pathway, then only the cell become die, something like that we have to prove. So we did so many molecular pathway to confirm facts analysis, other western blot or QRT-PCR. And also we do have some inflammatory effect. Like I said initially, if our inflammatory effect, anti-inflammatory effect goes down, if our immune system become weak, then we will high risk to get this kind of disease. So we do have confirmation with inflammatory pathway and inflammatory gene and protein to confirm with QRT-PCR and Western blot, and also other pathway detection. And yeah, this is the inflammatory cell to confirm accumulation of nanoparticle, and then some kind of comparison to recover. This is the normal cell to recover. The control cell, once we treated with some immune inducing some liposaccharide, when we add into the cell, it become highly produced immune system or immune cytokine, pro-inflammatory cytokine. This will be whether it is induced or not, something like that we confirm using this different type of stain, and also other molecular mechanism. And also sometimes we do have some non-target toxicity also, because this is a nematode. It's a highly sensitive, right? Even though we add very, very little amount, it will become very sensitive, and it will become, if our compound is toxic, it will be die, right? So we do confirmation with non-target organism to confirm it's a good, it's something like that. If it don't have it doesn't have the toxicity to the non-target organism or non-target cell, something that will be confirmed. And so many other molecular pathway. This is another nanoparticle which we have been synthesis using some kind of peptide to confirm anti-inflammatory and anti-cancer activity in the future. And also, not only in vitro, we do have confirmation with the animal mouse model. Then only it will be go to the next level. Even though we do so many molecular and so many things, we do have confirmation with a mouse model. Then after become animal model, then the third phase will be the human trial actually. So this is the drug, whatever drug we are having, we should undergo this all the steps. Then only we will get approved actually. So this is the process. And then sometime we produce some kind of photothermal therapy. This photothermal therapy, it will be very, very helpful because when, for example, you can imagine mouse have someplace tumor. For example, in the liver, the mouse has a tumor on the liver. Then we give drug to the mouse, right? Then we don't know whether the drug reaches exactly to the cancer population or cancer cell into the liver, right? They can be roaming around in the blood, right? After having the drug, we pass light directly to the liver, right? Then all the drug will come attract based upon the light. They can attract and all accumulated in exactly where the light passes. All will come into the exactly same place. Then they absorb the light and convert it into the heat. After the metal coated with our compound become very heat, then the cells automatically destroyed or died. This is the mechanism, is a target specific photothermal therapy. This called ferroptasis mechanism. That is like once it become heat, the cell become die because of that ferrous and ferric ion mechanism is called the ferroptasis mechanism. So that also we do check every time to have another alternative method. It's like a target specific method, one of the target specific method. And we also have some other pathway and so many things. And there are so many pathways these days we are focusing in our lab. And I think already I know that so many research faculty members, so many research scholars, and so many other highly motivated researchers are there in currently in the webinar. If anybody want to have more detail, actually I'm just passing very fast since we don't have time, less time. If someone want more detailed explanation, I do have complete complete molecular mechanism and how you can approach your research, something like that. This is the PA3 AKT pathway, is a post-translational pathway. Because post-translation pathway is very, very important pathway because this is the functional protein which is very, very important for cancer cell to further proliferation or further growth or further invasion or something like that. So we do check this kind of post-translation pathway through PA3 AKT pathway. And there's another vision of PA3 and protein ubiquitination. Protein ubiquitination is called some kind of unknown protein or unwanted protein in your body. So they will collect some unknown or unwanted protein. They, you know, right? Protein is made up of amino acid or peptide, right? They convert it to the simple peptide, a simple useful peptide. Just just like garbage, right? Just they collect all the unknown protein. They undergo some process. They convert it into simple amino acid or simple peptide. It will be really helpful for the new function. Actually, that is the process is called protein ubiquitination. And like I said, is autophagy phagocytosis mechanism. It's also one of the mechanism. You should have confirmation whether your nanoparticle can exactly following this pathway or apoptotic pathway or paraptotic pathway or so many other pathways. We do have confirmation before we go for the animal mouse model. So we should have this kind of molecular pathway. We usually do have, and MAPK pathway also we do have. This is the transcriptional pathway. That PA3 AKT is a translational pathway. Is a transcriptional pathway. There are two different pathways we do have. And finally, I would say that green synthesized or bacteria mediated synthesized nanoparticle will be very, very active to particularly to all type of cancer cell. And we do confirm with different molecular pathway method. And of course, we should have in vivo, and then only we could make us a commercial product finally. And this is the step we usually do. And also initially Professor Kalevan explained about there is some group in Korea. It's called Korea Tamil Nanbargal group. uh, I am putting a lot of career opportunity videos for students, for postdoctoral researchers and internship and so many things. If you have time, you can visit this YouTube channel, Korea Tamil Nanbargal in YouTube. You can see all other activities as well as your career opportunity, and everything is available in this channel. Yesterday we were talking some of the faculty members from this Sarada College Department, Zoology Department. I'm ready to have some kind of career opportunity for the student, because uh, I'm last 1 month I'm having different kind of webinar, and always say that Korea have a lot of opportunity for PhD, and particularly it's a 100% safest country for women. So no need to worry about that. Wherever you come to Korea, so we will come to know from this association. They have the member in each and every city, so no need to worry about your staying and everything. So there is a chance for PhD, there is a chance for postdoctoral, there is chance for master. You can come here. You can come here. You can do your own interesting topic as a PhD or postdoctor or intensive program, because why there is a lot of opportunity means Korean people, they don't want to continuously doing PhD, because they are going to, particularly the man, they should have a 2 year military, compulsory military camp. They should attend. They should go to military. Once they completed master, the age around 27 or 29, right? After that, they don't have interest, and they are thinking that they have to settle in the life, so they have to get married. They have to settle in the life, so they are not interested to join the PhD these days actually. So now all professors, they have the fund, and of course it's 100% scholarship. You no need to pay any tuition fee, and you will get a stipend and so many things which I have explained in the video. So you can see the video, you will get some idea. Please use this opportunity and make it use for your life and become more better. Particularly, like I said initially, from geology background, you should grab this kind of opportunity to success in your life. And I also personally will be very happy if you come to Korea. Please inform me anytime. And yeah, just I want to give some awareness to those who are in this webinar. Please do have try, and you should have this kind of opportunity to grab this kind of opportunity to succeed in your life. And thank you so much. Because of time congestion, I just make it more fast. Otherwise, I would have more detailed explanation. I hope you may understand this webinar. And if you have any doubt, if you have any clarification near future, you can contact me anytime. Okay, thank you. And finally, I would say that in Korea is a very, very beautiful country. We have 4 different weather seasons, like spring, autumn, winter, and summer. If you want to enjoy this season, you should come as a student or postdoc or your job related thing. You always welcome to Korea, and we are ready to welcome you also. Thank you so much to give me this opportunity, and I'm ready to take your questions and answers. Thank you.
Haribalan,
Thank you.
kindly provide your mail ID in the chat box.
Yes.
So participants, if you have any questions, directly mail to him, Dr. Haribalan. Thank you, Dr. Haribalan, for your enlightened speech on biomedical approaches of green synthesized bio-nanoparticles towards cancer therapy and inflammatory responses of cytokines. Thank you so much.
Thank you, ma'am. Thank you.
போடுங்க
Haribalan?
Yes, ma'am.
Change the host, Haribalan.
Okay.
Okay. Thank you, thank you.
சரி போடு தாயம் போடு.
சொல்ல வேண்டியதுதானே.
Now I invite Dr. A. Nirmala to introduce our next resource person. Participants, wait a minute.
சார் கலை சார் வந்துட்டாரா?
இல்லை இன்னும் வரல.
திரும்ப கால் பண்ணுங்க. இப்பதான்
ஃபோன் பண்ணிடுறேன். இது எப்ப அனுப்பிச்சிட்டோம்?
மதியானம் அனுப்பிச்சிட்டோம்.
எது?
ஹலோ.
அவரு வந்துட்டாரான்னு கேளுங்க.
வந்தாச்சுங்க, லைன்ல இருக்கேன்.
ஓகே சார், உள்ள வந்துட்டீங்களா சார்? சரி, ஓகே சார். சார் நேம் என்ன நேம்ல தருவீங்க?
Hello. Hello, participants. Sorry for the inconvenience. Now I invite Dr. A. Nirmala to introduce our next resource person. Participants, mute your audio.
ஓகே.
என்னப்பா அவ்வளவுதானா 10 நிமிஷம் தான்ப்பா வெளியே போய்
Nirmala.
ஹலோ.
சார் வந்துட்டாரா?
பாக்குறோம். அதான் ஏதோ கதிர்வேலன் சொல்லி பேர் வந்திருக்கான். கதிரவன் பாரு.
சார் நம்பர்ல இருக்காங்க சார். பாரு பேரு சார் பேர் ஒரு நம்பர் கதிரவன் சார்.
இருக்காரு
சார் இருக்கு.
இருக்காரு கலை.
ஓகேம்மா, யூ ப்ரோசீட்.
ஃபர்ஸ்ட் மாத்திட்டதுக்கு அப்புறம் நீங்க பிரசன்ட் பண்ண ஆரம்பிங்க. அது ஏன்னா சாருக்கு ஸ்கிரீன் ஷேர் ஆகாது. நாங்க ஹோஸ்ட் மாத்திட்டு உங்களுக்கு சொல்றோம் சார், அதுக்கப்புறம் நீங்க ஸ்கிரீன் ஷேர் பண்ணுங்க சார். ஓகே சார், ஓகே, தேங்க்யூ சார். அவருக்கு வந்து வீடியோ ஆன் பண்ண சொல்லுங்க.
கலை அவதானே பண்ணணும்.
கலை தான் பண்ணணும். ஃபோன் பண்ணுமா கலை?
Good evening to another person here. Our resource person, Dr. K. Kadiravan, completed his PhD in Bharatidasan University in the year 1996. 1997 to 2004, Sir worked as a scientist in the Entomology Research Institute, Loyola College, Chennai. Department of Science and Technology sponsored BICAS Fellowship, and he went to Israel during the year 2000 to 2001. 2004 onwards, he started his career in the University of Madras. 2008 to 2009, a resource person received DBT Overseas Fellowship. In addition to that, he published 75 papers in the national and international journals. In the past 10 years, our resource person has received 1 crore as grants from government bodies such as DBT, DST, UGC, MHRD for his research. Currently, he has 2 DST projects in his hand. Our resource person has produced 5 PhD students. Now, under his guidance, 7 PhD scholars are going to pursuing PhD. His current area of research is plant virology and nanotechnology. Sir has visited several abroad countries like Israel, UK, and Malaysia. He is a sincere, devoted, ever-helping, eminent professor. We are really fortunate to have a person like you, sir. Now I invite our resource person, Dr. K. Kadiravan, to take over the session.
Hello, Kadravan sir.
Sir?
Hello, sir.
Kadiravan, sir?
1 minute, 1 minute. அவர் ஹோஸ்ட் அப் ஆயிடுச்சா? நிர்மலா
சொல்லுக்கலே.
ஒன்ல அவருக்கு இது அன்மியூட் பண்ண சொல்லு. அவர் ஹோஸ்டா சேஞ்ச் பண்ணிட்டேன், அவர் அன்மியூட் பண்ணாம இருக்காரு. கொஞ்சம் கால் பண்ணி சொல்லு. ஓகே.
அவருக்கு அன்மியூட் பண்ணலையா? அவர் ஹோஸ்டா மாத்தியாச்சாம். அன்மியூட் பண்ணலையா? அவர் அன்மியூட் பண்ணாமலே இருக்காங்க.
Hello, now can you hear me, ma'am?
Yes, sir. Yes, sir. Audible, sir.
Yeah. Can you share the share screen?
Okay, sir.
Hello, Kadravan sir.
Yes, ma'am.
You are the host. You can share, sir.
Okay.
Any unwanted noise, you can mute, sir.
Okay, ma'am. Okay.
Any participants, any unwanted news?
Okay.
Okay, sir. Okay.
Sir, make it as a full screen, sir.
Okay. Now it is okay, no, ma'am?
Okay, sir. Okay, sir. Now okay, sir.
So once again, good evening to everyone. I must thank for the organizers, Dr. Nirmala and Kalaivani and head of the department and the principals of the Sarada College. So this is the opportunity for me to brief and tell me about the viruses. So in our laboratory past decades, we are working in the viruses, particularly in the plant viruses. So at this juncture, in the pandemic conditions, everyone know about the viruses, but we should know about the basic concept of the viruses. That is why I give the topic knowledge of viruses during the pandemic conditions. So what is viruses? So before going to further this one, what is viruses? So we must know, everyone know the virus has one of the major characteristics that they are obligate intercellular parasites. So viruses are unable to grow and reproduce outside the living cells, we know, because all the viruses need the host. So, and another one is this: no virus is able to produce its own energy. So always the virus is depend on the host machineries. That is the thing. And another one important thing is the virus is the nanoparticle. So nowadays We know about the nanoparticle, silver, gold, polymer, several nanoparticles they are used for the different purposes, even though for the medical applications, for the drug delivery and other things. So the same time, the virus also used as the nanoparticle for the drug delivery system. That is the new trend is going on. So virus is often described as the nucleic acid surrounded by proteins. So is found in all the living organisms. Virus is found in all the living organisms, including the animal, plant, bacteria, fungi, soil, marine sediment, and seawater. Even the seawater, we know the white spot syndrome in plant culture. The white spot syndrome is carried by the seawater. See the fungus, everything. So all the organism is caused by the viruses. So virus is nothing is there. Virus particle is consist of only either 2 or 3 parts. There is no other material is there. So the genetic material, either the DNA or RNA molecules, is carried the information. These genetic molecules is covered with the coat particles, coat proteins. So sometimes some of the virus is contain the envelope, some of them is the lipids. So that is the things. So the envelope of lipid that surround the protein coat when is the outside of the cells. So the size of the viruses, it will be, so these figures shows the size of the viruses. So in the human being, we know that is the 1.7 meter or some things. Okay, but in small molecules, 10 to the power of 9 is the 1 nanometer or proteins. So the plant cell is 10 to the power of 4, animal cell is 10 to the power of 5, and bacteria is 10 to the power of 6. It means 1 micron millimeter. But in case of the viruses, it is the less than 100 nanometers. So we have seen only through the light electron microscopes. Otherwise, you can't see the things. So these are the viruses classified in the different category, particularly in the 7 category. So that is called the double strand DNA virus, single strand DNA virus, single strand RNA virus, negative.
Hello, Kadravan sir.
Yes, ma'am.
Please mute Sujata and Dina Dayalamurthy sir.
Just a second. Yeah, I couldn't find out this. Unmute all.
No sir, no sir.
No?
Just mute them.
Sujata.
Sir, give mute all, then you can unmute.
Click on the button to free webinar.
இங்க ஒரு மாடு அம்மான்னு கத்துது இந்த வீடியோல.
Hello sir.
Yes ma'am.
Otherwise change the host sir, I will do.
Okay, how can I change the host?
So in your name next there is a more option sir. Near to your name.
Okay, just a second. Yes, I am not familiar with this one. Okay.
Mute him or mute it.
Participants, mute yourself. Please, participants,
Mute
mute
it.
yourself.
Unmute it.
Hello, participants, mute
Please
yourself.
mute yourself.
Unmute it.
சார் உங்க நேமுக்கு பக்கத்துல பாருங்க சார், மோர்னு ஆப்ஷன் வருதான்னு பாருங்க. மியூட் அன்மியூட் அதுக்கப்புறம் மோர்னு வரும் பாருங்க.
மியூட் மியூட் ஆல் அன்மியூட்னு இருக்கு மேம், அந்த இது இருக்கு.
மோர் அப்படிங்கிற ஆப்ஷன் வருதுங்களா?
ஆமா, மோர் இருக்கு.
மோர் கிளிக் பண்ணுங்க.
ஓகே.
அதுல சேஞ்ச் ஹோஸ்ட் வரும் பாருங்க.
ஆட் ப்ரொஃபைல் பிக்சர் ரீநேம் தான் வருது. ஹோஸ்ட் பக்கத்துல மோர், அதுல வந்து உங்க தீனதயாள மூர்த்திங்கிற ஒரு நேம் இருக்கு.
1 minute sir.
95 participants in the meeting.
Okay sir, 1 minute sir, 1 minute.
Dear participants, please mute yourself. Dear participants, please mute. Then only we can continue our class.
Sir, options வருதுங்களா?
ஆப்ஷன்ஸ் எனக்கு வந்து அந்த இதுதான் மேம் வருது, மோர்ல தான் வருது.
மோர் கிளிக் பண்ணுங்க சார். மோர் கிளிக் பண்ணா என்ன வருதுன்னு சொல்லுங்க.
ரீநேம், ஆட் ப்ரொஃபைல் பிக்சர்ஸ்னு வருது. இல்லன்னா பக்கத்துலயே மியூட் அண்ட் மோர்னு இருக்கு.
அந்த மியூட் கொடுங்க சார். மியூட்.
சார் ஒரு நிமிஷம் சார், ஒரு நிமிஷம் சார். கதிரவன் சார்.
எஸ் மேம்.
கலைவாணி பாலசுப்ரமணியன் இருக்குங்க சார். அந்த நேம்ல போயிட்டு பண்ணுங்க சார். அதாவது மேக் போஸ்ட் வருதானே?
கலைவாணி பாலசுப்ரமணியம் அங்க வந்து
மேக் போஸ்ட் வருதான்னு பாருங்க சார். அங்கதான் மோர்ல கிளிக் பண்ணா வருதா பாருங்க.
அங்க வருது மேம்.
ஓகே, அங்க கொடுத்துருங்க.
மேக் ஹோஸ்ட்
கொடுங்க சார்.
கொடுத்துருங்க சார்.
கொடுத்துட்டேன்.
வந்துருச்சு மேம்.
கலை நீ பாத்துக்கோ இல்லப்பா.
ஓகே. Kadravan sir?
Yes ma'am.
You can proceed sir. I will share the slide.
Okay, there is no issue ma'am.
Okay sir.
Okay, so the size of the viruses, it will be range is a nanometer. So based on the classification, the virus is classified different aspects. It's the double strand DNA or single strand DNA. Sometimes it's a single strand RNA, positive strand, negative strand, and double strand RNA. So like that, it will be vary. So the virus particle is based on the structure. It is classified based on the basiliform, spherical, filamentous, Gemini viridae, and rigid rod shape structure is present. So what are the viruses? So it is observed only in the electron microscope. So it is composed of the code made up of protein, sometimes a lipid wall and a nucleic acid core. The genome of the viruses, all the viruses, even the animal viruses or human viruses or plant viruses, is encoded in the 3 or more than proteins, more proteins. Each protein have their own function for the different aspects. So the genome is translated to produce a protein. So the DNA, central dogma system, the DNA to RNA, RNA to proteins. So the nucleic acid genome is replicated within a cell, producing nearly many as 1 million copies. You can imagine that. So the virus is required the host cell for multiplication. Without the host cell, it won't be multiplication because the virus doesn't have the replication mechanism. And this virus cannot function the outside the host cells. Cannot function the outside the host cells. But the particle of virus can be stable for many years. So here it is the important thing. The virus cannot function the outside the host cell, but it can be stable for many years in outside the host, either the debris, water, or soil, or air. It will be survive, but it won't be multiplication. So the viral genomes are structurally highly diverse. You know that even the RNA viruses have the single standard, sometimes it's the double standard, sometimes it's the linear things, some of them is the circular. So same time the DNA also, some of the DNA virus are the single strand, some of them is a double strand. So some of the single strand DNA virus is the positive, some of them is the negative. So like that, the genome structure is varied. So according to the Baltimore classifications, so the virus are classified based on the replication strategies and as well as the genetic content, as well as the genetic content, as well as the replications. So Baltimore classified in nearly 7 classes. So that is the first double strand DNA viruses. So the best example is the adenoviruses, RCA viruses, poxviruses. Then second one is the single strand DNA virus, is the positive strand, or is the sense oriented. Sense oriented DNA. Then third one is the double strand RNA virus, that is called, the example is the Rio viruses. Then fourth one is the single strand positive viruses. Single strand positive viruses. Then single strand RNA viruses in negative strand or the antisense oriented. Then single strand reverse transcriptional strand. This is a strand or sense RNA with the DNA. So here it is the single strand RNA is cover intermediate with the DNA. Intermediate with the DNA. That is called the single strand RNA viruses and double strand DNA RNA viruses. So according to the Baltimore is classified all the viruses, including the plant, animal, and everything. So here it is the viruses. So the classification, the double strand DNA virus is converted to the mRNA, then mRNA to the proteins. Like that, the single strand DNA is synthesized the double strand DNA. Again, it will go for the mRNA. But in case of the double strand DNA, there is no need to go for the DNA. Directly, it will go for the mRNA and it synthesizes the protein. So in case of the single strand DNA positive strand, they again synthesize the DNA RNA hybrid. So they produce the double strand DNA. So the single strand RNA positive strand synthesize the negative strand, then it go for the double strand DNA, then it is go for the mRNA, and finally it go for the protein strand. Like that, double strand DNA is the single strand RNA and the mRNA and protein strand. Like that, it is a classified the Baltimore. So now we go for the mechanism, how viruses are infected with the cells. There are the magic 6 major steps. These major 6 steps is common for all the viruses, but all the viruses it will be differ. But most of the viruses is following this kind of replication and infections. The first thing is the absorption. Virus bind to the host cells. In case of the animals and other things, virus attract the receptors. Okay, so it is attached attachment is the very important. So attachment to the adjacent receptors. But in case of the plant cell and other thing, some of the virus is attached in the wounding regions. So that is in the plant cells. Some of the viruses are transmitted through the seeds. That is also in plant viruses. But in case of the animal viruses, so this attachment is attached in the receptors, binding receptors. So here it is the binding with the specific receptor. Each one is differed. But in case of the COVID-19, the spike protein is attached to the receptors. So we will discuss in the later. So this is the attachment. Once it is attached in the surface of the cell and it is the receptors, it is penetrate to the cells. So sometimes the whole protein complex is entered and it will enter to the cells. But in some cases of the plant cells, only the nucleic acid is penetrating the cytoplasmic. So it will be vary from the some of species to species. So the penetration is the second step. So the virus inject in the genome into the host cell. Sometimes the whole viruses, including the protein body, will enter to the host cells. So once it will enter to the host cells, the genome get replicated. So we know that is the virus doesn't have the mechanism, replication mechanism, means the DNA dependent polymerase or the RNA dependent polymerase doesn't have the viruses, because the virus doesn't have, nothing is there. The virus is genome, the genetic material is covered with the protein molecule, code protein, nothing is there. That is why it won't be produced the ATP molecule. So for the mechanism, for the replication mechanism, for the replication, virus replication, so always depend the host mechanisms. So in the host country have the replication, no, the DNA dependent polymerase or RNA dependent. These polymerase, the enzyme utilized the virus and it will be replicated in the host cell itself. So that is the replication mechanism. So it is purely utilize the replication machinery from the host materials. Then once the replication is over, then we can go for the assembles, because the assemble is very important. Assemble means the replication, the genetic material is replicated, then after that the protein molecules is assembled. So that is the fourth step. Then fifth step is the maturation. Full length viral DNA or genome or the DNA genome is a viral component, the genetic component is fully developed and the protein code also is developed. Then once the things, it will be released, released, expelled from the host cell, and it will be penetrated to the uninfected cells. One cell to another cell, it will be penetrated. So this is the normal mechanism of the viruses in all the virus species. Virus types by nucleic acid composition, we know that is the DNA virus and the RNA virus. The DNA is the single strand DNA virus. There is no envelope. There is no envelope. But in case of the some of the single strand virus, naked is 5 nakeds and 100 species is available. In double strand DNA viruses, 9 enveloped, 12 naked, and 200 species in enveloped, and 300 species are naked. Like that in RNA virus also, the single strand RNA is 9 enveloped and 200 species, and 14 naked families and 600 species is there. Then double strand RNA also 2 enveloped families and 5 naked families based on this. So these virus type is both the DNA and the RNA wise infected in all the vertebrata, invertebrata, plant, fungus, and bacteria. So invariably, this DNA and RNA virus is affected in all the living organisms. So these are the simple replications. So these are the 7 Baltimore classification in one slide. Okay, so the virus DNA. So viral DNA, it will be progeny of DNA, progeny of virus and protein, or it is the mRNA, the early protein, late proteins. So like that, the single strand DNA, virus DNA is single strand DNA, the genome is single strand DNA. So these single strand DNA synthesize the double strand DNA, Then it will go for the mRNA. Then is the virus protein. Then the virus, once the virus protein is formed, then progeny of the virus is released to one cell to another cell. Like that, the double strand RNA. So the virus RNA directly go for the mRNA, then particle assembly and progeny. Like that, in the virus RNA, positive strand, single strand RNA is they synthesize the minus strand RNA, then is the progeny of the viruses. Like that, the Baltimore classifications is the 7 classifications is there. So virus is spread in the many ways, either the plant or the animal or the human. So mostly virus or plant often transmitted from plant to plant by insect. So these viruses need the vector that is called the insects that feed on plant sap, such as the aphids, like that. Viruses in animals can carry by blood-sucking insect. Viruses in animals is transmitted through the blood-sucking insect, like a mosquito and other things. So these viruses bearing organism are known as the vectors. So the influenza viruses are spread by coughing and sneezing, like a COVID. So the range of host cells of that virus can infect is called the host range. So this can be narrow. Maybe this is a specific host. Or sometimes when a virus is capable of infecting many species also. Okay. Mostly the virus are the host specific. In some time it will be infecting the several species. So now coming to the second part of our discussion is the plant viruses, and third part is the COVID. So the second part is the plant viruses. Our laboratory is the last with the 10 years. We are from 2008 onwards. We are working in the plant viruses. So the plant viruses is one of the very important problem symptoms, the plant viruses, because it is a symptom is more or like a symptom. Based on the symptom, it is very difficult to distinguish the viruses, because even the bacteria, even the fungus, even the nutrient deficiency, the same symptom will appear in the plant. So it is very difficult to distinguish between the bacteria or the nutrition symptom or the viral symptom. So that is why it is very difficult to work on this plant virology. Same time, the complication of the viruses, because we know the 7 classifications. So some of the viruses are DNA viruses, some of them is the RNA viruses. So we need some very good skill trainer, skill technician is the very important. So sometimes the symptoms may also be helpful in identifying the viruses in causing diseases. Sometimes, not 100 times, 100 pure. So sometimes it will be helpful. So what are the symptoms is affected? So these are the symptoms is affected. More than 2,000 type of viral disease of plants are known and cause spots on leaves, fruits, stunted growth, and damaged flowers or roots. So the most of the plant viruses have the RNA genomes, but the DNA virus is the very important scenario, significant damage to the plant. So these are the plant viruses spread disease in 2 major modes, that is the horizontal transmission and the vertical transmission. So in case of the horizontal transmission, is entering through the damaged cell walls. So in the farm field, in the cucumber or tomato or brinjal or anything, bendi or like that. So so most of the RNA virus is transmitted through the horizontal transmission. So whenever whatever the tools we are used in the farm, so the from the tools one plant to another plant, the virus is carried by the tools. Same time, the vertical transmission, this vertical transmission inheriting the virus from a parent, means the seed transmission. So the virus is infected from the seeds. So that is a 2 type of diseases. So these are the plant virus symptoms. So stunting is the common, mosaic symptoms, ring spot, abnormal growth, tumor, enasion, blights, wilt, systematic necrotic lesions. These are the symptoms. One by one, we can see the another slide. So these are the things. Chlorosis. So these chlorosis is due to the virus problem, but we don't know. In the farmers, they don't know about the viruses. So this the symptom is similar to the nutrition deficiency. Also is there, but the chlorosis is due to the viruses. So the yellowing of plant parts, leaves, fruits, flowers is getting yellowing. So the another one types of symptom is the mosaic symptoms. See, these are the mosaic symptoms is due to the viruses. But even the some of the insects, aphids, mealybug, these are the organism, insect also cause this kind of symptoms. So that is why I am telling this is a difficult to distinguish, difficult to identify the virus symptoms. So here it is the vein banding pattern. So the vein banding pattern is vary. Vein banding pattern is vary because of these viruses. Chlorosis, yellowing of tissues, means this vein is affected by the viruses. But the same symptom appeared in the mealybug also, is created by the mealybug also. Even the white thing, white fly also. So that is why. So this necrosis, the dark color spot is appeared. Dark color spot is appeared. That is the necrosis appeared in the leaf, even the fruit also appear. Plants have. So this this is the due to the viruses, but this kind of problem is this kind of dark spot is created or caused by the bacteria also, fungus also. So that is why it is difficult. Then the mosaic symptoms. The mosaic symptom is appeared in the tomato fruit, even the tomato leaf. So these are the problem is created. So these are the mosaic symptoms is purely by the viruses, but the pharmacist doesn't know about this one. The leaf rolling and curling. So that is also is caused by the viruses, but these are the symptom is sometimes this symptom was caused by the animal also. The stunting growth. Some of the plants, if you visited the farms, these are the chili plant. Chili plant, chili field. If you see the chili plant, some of the plant is getting dark, stunting growth. So this stunting growth is purely in the viruses. So either the root or leaf or stem is getting stunted. So the small leaf is appeared. Is not a normal leaf is appeared. So this is also is created by the viruses. Sometimes the yield loss. Yield loss. Okay, yield loss. The potato is the major problem. Potato spring spot viruses is the major problem of potato. So the yield is very low. So the yield is very low. Then tuber also is getting this kind of symptom. Ring spot rings is appeared. That is also in the viruses problem. Sometimes the stem is getting pearled. Loss of woody perennials. Okay, the stem is getting affected. But this is also the virus problems. The blight diseases, the entire leaf of the top of the regions is getting blight. So like a yellowish color. So there is no chlorophyll. So that is as a blight also is caused by the viruses. And wilt, the top of the plant, particularly the watermelon plant. Watermelon, most of the watermelon is affected by the wilt disease. The tip of the top of the plant, the tip is getting stunted. So it's getting brown. The leaf is, there is no chlorophyll. So that is causes also is the due to the viruses. So these are the symptoms is based on the viruses. So according to our laboratory, we are working in the DNA viruses because we know that is the DNA and the RNA viruses, because we are not concentrating on the RNA virus, because the DNA virus is one of the important virus in throughout the world, not only the India, not only the Tamil Nadu, entire world is affected by the DNA viruses. Because why I am telling the DNA virus is the important, because this virus is carried by the vector. So these are the vector, the leaf hooper, tree hooper, white fly. These are the organisms, aphids, mealybug. These are all the insects. Is carry the viruses. Is these are the insect is transmitted to the one plant to another plant. These are the vector. It's transmitted one continent to another continent. Like that the leaf hooper. Nowadays we are facing the problem in Rajasthan, Delhi, and those areas are facing the problem. No, the green hooper. So migrated from the Pakistan, Afghanistan. Like that, these are the vector is migrated from India to Pakistan, Pakistan to Afghanistan, Afghanistan to the Middle East, Middle East to Europe, and Europe to Africa. So throughout the world, it is migrated. It is carry the virus also. That is way. It is the one of the important viruses. That is why our laboratory is working on this vector, the species virus, particularly family of Gemini viridae. So the generally called Gemini virus, the family called Gemini viridae. So this Gemini viridae is classified the four genus like a master virus, virus, cartovirus, and Bergamo viruses. So based on the genome organization, vector are things, and the plants based on the hospital, these four species is classified. So some of the master virus is the single genome. Virus also is a genome virus, and virus also single genome. So already I told this is the DNA viruses. This DNA virus is single strand DNA virus. That is the speciality for the Gemini viridae. Gemini viridae is a speciality, is the DNA virus and the single stranded DNA viruses. So these three master virus, virus, and the cartovirus is the single genome, and the Bergamo virus is the double genome. So the virus containing the genome A and the genome B, both the genome are the circular DNA. So these viruses infected in the carried by the transmitted by the specific vector. Okay, particularly master virus is transmitted by the leaf hooper alone. This tobacco virus is carried out by the tree hooper. Then virus is the leaf hooper, and the Bergamo virus is the white fly. So specific vector is carried. So and based on the plant, master virus is affected only monocot, only the wheat or rice like that is affected. But all the other three, virus, Bergamovirus, and virus is affected in all the dicotyledon plants. So among the four species, our laboratory is specifically working on the Bergamo virus. Because these Bergamo viruses is infected, these Bergamo viruses is affected in all the vegetable crops. Whatever we are using in the vegetable in our kitchen is affected by the Bergamo viruses. So we can one by one. So these are the biological properties, the insects, aphids, whitefly, hooper, thrips, plasmodiaphoris, mites, and nematodes is carry the viruses. So that is the vectors. Okay. So important viral disease, the what are the vegetables. So these are the vegetable is affected by the Bergamo virus. So bendy, whatever the ventacle is the yellow vein mosaic viruses is affected by 90% yield. So once the virus is affected in the bendy field, the total crop yield is lost nearly 90%. Cardamom, whatever the Western Ghats, most of the cardamom is affected by the mosaic symptoms, mosaic virus. Cardamom mosaic viruses is 70% yield loss. In the groundnut, groundnut butt necrosis is one of the important viruses affected. it is yield loss is 80%. Whatever the pulses we are used, green gram or mung gram or any grams, okay, is the yellow mosaic virus, nearly 70% yield loss. In potato is the potato mosaic viruses is nearly 80% yield loss. In tomato, leaf curl diseases is 100% yield loss. You can imagine. So once the farmer is initiated the seeds germination, after the fertilization, water, the growing. So at the time of 3 months, the entire 3 months work is totally spoiled by this tomato yellow leaf curl virus. Like a chili leaf also is the same thing. Chili also is 100% yield loss. So at the time of flowering, tomato or the chili, the virus is entered and spoil the entire area, either 3 acres or 5 acres. It will be spoil everything. Even you can't get the single fruit of tomato or single fruit of the chili, melaga. Nothing will be there. So the total input for the 3 months of the input, the total completely waste. So that is the major problem. So this is the germination. So the bergamot virus is affected only the dicotyledon plants. It attack the tomato, bean, squash, and cassava and cotton. So there are currently 424 species in this genus, including the type of bean golden yellow mosaic viruses. So already I told that is the bipartite genome A and genome B. Two genome is appeared. Some of them is the monopartite. This virus, that is a complication. The people are not ready to work in this kind of viruses because some of the viruses is virus. The same virus is a single genome, monopartite. Some of them is the bipartite, means the two viruses, DNA A and DNA B in same viruses. So this virus is around 3000 base per size. So you can imagine 3000 base per both the viruses. So each virus have the, already I told, 3 to more proteins. Like that here the several proteins AV2, AV1, AC3, AC2, AC1, AC4, like that. Different based on the species to species, these protein is, it will be vary. But in bergamot virus, these are the protein. So another one interesting is that some of the protein are the sense oriented and some of them is the anti-sense oriented. See here the AV2 is sense oriented and the AV1, that is the core protein, is the sense oriented, but in the AC3 is the anti-sense oriented. AC1 also is anti-1 oriented. This is the replication things. AC4 is the anti-oriented, anti-sense oriented. So these are the virus is the sense oriented. Some of them is the anti-sense oriented conditions. So this is the same thing applicable to all the viruses also, not only not for the human viruses or the things. Everything is the same. Some of them is oriented, sense oriented. Some of them is the anti-sense oriented. Another one interesting is that these viruses are in the overlapping also. See here, the AC2 is overlapped with the AC3, and another one thing, AC2 is overlapped with the AC1. So this protein is overlapped with the another one protein. So that is also is the interaction. So these are the IR region is the common region for the, all the Gemini viruses. So these are the base pairs from the 300 or 400 base pair is common for the all the Gemini viruses. So if you want to identify the Gemini viruses, to confirm the Gemini viruses, you can use this kind of, use it from this primer, the forward, and reverse primers from the intergenic regions. If you isolate and you can go for the amplification, definitely you will get the amplification, means that is the Gemini viruses. So this region is the common region. Another one interesting thing is that here the sum of the region, the particular region is continuously mutated. That is a very important one. So that is why the virus strain is vary from species to species. Whenever the replication is appeared, the virus also is getting mutated. So why I am telling now, here the genome size is very less, means that is just 3000, less than 3000 base pair. Among the 3000 base pair, this portion is just 300 base pair or 400 base pair regions. These 400 base pair is continuously mutated either by the natural or replications. Natural replications is continuously mutated. Mutated means not the base pair change him for the 100 base pair or 300 base pair change. Here just 3 base pair or 4 base pair is mutated or is changed. That is the new strain. So if you, so there is, because the genome size is very less, even though that particular region of the mutation region, continuously mutant regions, there is no other area will be mutated. Only the particular area, particular segment of the protein is getting mutated. So either the 4 base pair or 5 base pair is mutated. That is the new strain. So that is the our reporter is that. So these are the gene segment. That is the V1, V2, CL, C1, C2, C3, C4. This each one of the protein has the different responsibility. So the V1 is the code protein, V2 is the movement protein, and the C1 is the replication initiation. So this fragment of the protein is responsible for the replication. This fragment of the protein is responsible for the transcription activator. Then this C3 is the replication enhancer. So this is the replication. So these are the C4 is the determine the symptom. So that is the very important. These are the particular protein is determine the symptom of expression. If you change anything, base pair is change in this particular region, the symptom will not appear. So that is where the COVID, no, some of the people getting the symptoms, some of them is the asymptomatic conditions. So these are the protein, the specific proteins. Already the plant protein is identified, but in COVID protein, symptomatic proteins, till now the work is going on. So the C4 is the responsible for the symptom creator, symptom expressions. But the some of the protein or some of the C4 is not properly working, the symptom will not appear. But the virus is enter, virus is multiply, everything is there. Some of the so already I told bipartite genome. So the another one parted is the segment B. This is also is a circular DNA. So this is also same 3000 base pair, but the function is very less. So nuclear shuttle protein and movement protein. So the movement protein is movement of the segment B to the another viruses. So that is the thing. So each protein have the different role for the symptom creation, replication, transcription, everything. Okay, so these are, if you lineage the viruses, the DNA, Bergamo virus, there is a common region, I told you, that is the IR regions. So the AV1 region is the biggest one, AC2, AC3, AC4, and AC1. So some of them is the sense oriented, some of them is the anti-sense oriented, and interlocking also. Okay, so it is the sense and anti-sense is interlocking. So that is the thing is that. So these are the major vegetables what we are consumed in the kitchen. So these are the things is affected by the viruses, particularly the Bergamo viruses. So these Bergamo viruses, already I told, that is the carried by the vectors. So this Bergamo virus is carried by the white fly. So the cassava is the one of the plant is affected by the Bergamo viruses. So see the cassava mosaic viruses. So the mosaic symptom was appeared in the leaf. So once the mosaic symptom is appeared, the leaf is get curled. So ultimately the chlorophyll synthesis is less. So ultimately the yield of the tuber also is reduced. So nearly in the South Africa, nearly 90% of the 100% yield loss was due to the cassava mosaic viruses. Another one thing we know, that is the Solanaceae plants. All the Solanaceae plants, potato, tomato, tobacco, pepper. all the things, all the solanaceae plants is affected in the bergamot viruses. Even 100% yield loss is appeared. See the flowers. Once the flower is initiated, the white fly is enter, white fly is carries the viruses and it's affecting the plant. So that is the thing, sir. Then Wakra, Bagama virus, Bendi, we know the Vendaka. So this bendy plant is affected by the bendy yellow vein mosaic viruses. You know that these are the mosaic symptoms. Is getting curled, the leaf is getting curled. So ultimately what happens? The fruits also getting small. So the consumer is not attracted by these fruits. So this is also is a major problem. And Bergamo virus not only the vegetable crops, even the flower plants in the jasmine. Jasmine flowers getting the mosaic symptom, the yield is lost. So these kind of fruits, flowers is affected by the ornamental crops also is affected by the things. All the leguminous plants, all the legume plants, cowpea, mungrams, okay, green grams. So all the plants, legumination plant is affected by these viruses. So yield loss. So cooker beets, not only the cooker beet, sponge gourd, pikanga, ridge gourd, okay, soraka, pumpkin, then ridge gourd, bitter gourd, pavaka, cucumber, all the cooker beet is affected by these DNA viruses, that is called the Bergamo viruses. Not only the Tamil Nadu, entire world is these Bergamo virus affected all the plants. So that is the thing, sir. So the tomatoes, we know the tomatoes, the entire leaf is getting curled. Then how can we get the fruits from this plant? So entire plants, see the size of the plant at the just 2 months, just 2 months of the plant before giving the flower, the plant is getting curled. Means there is no flowers, there is no fruits. So nothing you will give. 100% yield loss in tomato. Even the papaya. So the papaya, we know that each place, each our garden, we saw that this kind of curling. Once the papaya ring spot virus is affected in the papaya plant, so the entire leaf, top of the plant, leaf is tip is getting curled. Once it is curled, the fruit size is very reduced and simultaneously there is no yield. So 100% yield loss. In most of the Coimbatore district and the Erode district is affected by this papaya ring spot viruses. So this is the problem major things. Then chili, so entire India, even the Andhra Pradesh, Tamil Nadu, everywhere, the entire area is Athur and Perambalur. These are the districts. They are cultivating the chili plant. So all the chili plants, nothing is there. Nothing. There is no flower. There is no fruit. Nothing is there. So everything is getting cut and simultaneously 100% yield loss. Even a single fruit will not produce this kind of problems. So not only the vegetable crops or ornamental crops, this Bergamo virus is affected the weed also. We know the Abatulan India, that is the Kupameni. So the most of the Kupameni is affected by the viruses, Bergamo viruses. We in the college side or in the road side, we saw the Abutilan India, that is a Kupameni. Most of the Kupameni is color is different, yellow is color. So that yellow is color means that is the virus is infected. So why I am telling the wheat also is affected by the viruses. The same tomato yellow leaf curl virus or the chili leaf curl virus or bendy yellow mosaic virus is affected by the weed plants. Once the harvesting over in the field, once the tomato harvesting over, what we will do? We will destroy the tomato field. We will destroy the tomato plant, but not in the weed. So once we replanted the tomato, the virus is came from this weed and is attacked the things. So our farmers, our Tamil Nadu, our Indian farmers, we are not a big farmer. Within a acre, 1 acre, we are cultivated the 1 row tomato, brinjal, 1 row pulses. Like that, we are cultivating, and we are cultivating also is the alternative crop system, sir. One season we are cultivating the tomato, then another season we are cultivating the rice, then second season we are cultivating the tomato. So we are alternatively planted the plant things, vegetable crops. But what happened whenever we are using the tomato, the virus is entered. So how come the virus is entered? How come the virus is sent? Know the tomato plants. So what we are saying, what I am trying to saying, these are the weed plants carry the vector viruses. Is the reservoir for the viruses. So whenever the optimal condition is arise, this virus is transferred to the weed to the vegetable crops. So that is why our recommendation to the farmers. So you must destroy the weed plant also. Okay. So whenever you harvesting is over, you go for the destroy the whole vegetable plant. Simultaneously, you can destroy the weed also. That is our recommendation, sir. So particularly these are the white flies. Okay, these are the white flies, particularly the sprilling white flies. That is a sweet potato white fly. That is called the Bemisia tabaci. These are the white fly is act as the vector to carry the Bergamo viruses, carry the Gemini viruses to the one plant to another plant. So it is very difficult to control the viruses, but it is possible to control the white fly. So this is the Bemisia tabaci is the act as the vector. It is the one of the very important things. So these are the life cycle of the Bemisia tabaci. So after the adult white fly, so it is the each adult is nearly 18 to 28 to 30 adults. Egg is laid, and each egg is to the complete the life cycle is 18 to 28 days. 18 to 28 days, that is the life cycle based on the temperature regions. If in case of the low temperature, the cycle it will be very long. In case of the normal temperature, the 38 degrees centigrade in the summer season, the temperature, the life cycle is very short, like this. So once the female can lay more than 8 per day, and between 150 to 300 in a lifetime of the egg. So each egg is multiplying with things. So the number of eggs laid by day vary with the temperature and suitability of the host plant. Here I am trying to say the white fly movement is based on the temperature and wind blow conditions. So the wind, wherever the wind direction, so the same direction the white fly also movement. So the based on the monsoon, based on the wind direction, the virus is also is moved from one place to another place or one district to another district based on the temperature and the wind blow. So these white fly can also damage the plants. Okay, not only the virus is damaged, the white fly also can damage the plants. Is the direct damage is when at the time of feeding of the plant, so it will disturb the midrib and other time. And by the indirect damage following the segregation of sugars, the honeydew is secreted by the white fly. That is why it is also is a problem. Then physiological disorders is also is appeared. Then transmitting plant damaging the viruses. So these are the damages is caused by the white fly, not only by the vector viruses. Okay, virus is also is damaging the plant, and white fly also is damaging the plants. So that is the feed due to the feeding damages. These are the deformation and defoliation, and finally the plant is dead due to this white fly. Not only the viruses, and the some of the honeydew is secreted in the plant. So sticky honeydew is secreted by the white fly, and it is appeared in the fruits. So this the black shooting mold fungus is attracted by the honeydew, and it is grow in the plant, so it is not attracted by the consumers. So that is also one of the problem. So here, one of the interesting thing is the virus transmission. How the virus is transmitted in the vectors. That is the one of the things. How the Bemisia tabaci transmitted many viruses, especially the Gemini virus. So whatever the insects, either the leaf hooper or the anything or the larvae, Spodoptera or Helianthus, all the organisms have the digestive enzymes. Whatever they consume, it will be digested by their own enzymes. But here, the protein, the virus is entered to the whitefly, and it will be transmitted to the another plant. So means this virus is protected by the whitefly. So that is a very important thing, Sam. So here the circulatory route of virus whitefly. So the stylet is observed. Stylet is the stylet is penetrate to the plant, and it will be enter to the xylem phloem of the plant, and the stylet is suck the phloem and xylem, the phloem sap. So suck the phloem sap, and simultaneously the stylet is absorb the virus particle also, and it will be transferred to the uh, transmitted to the salivary gland, and digested tract, external digestive tract, enter to the digestive tract and mid gut, salivary gland. Again, it will be settled in the saliva. So means here whatever the digestive enzymes here, gut membrane is not affected as are not inhibited the viruses. So some of the special protein is covered or protected this protein. That is why the virus is entered and it will be rejected. Okay, otherwise, other whatever the foreign material, it is digested by the white fly. But except this virus particle is not digested because of that proteins. So that is the thing. So that is why this is the very important things. This is the plant viruses is very important, particularly this DNA virus, single strand DNA virus. The size is nearly 3000 base per. Okay, it will be it is affected in the all the vegetable crops in the throughout the world. It is movement also. It is the very easy because there is no barrier. It won't be controlled. So, but the several mechanism to control the viruses, but nothing is there. Only thing we have to control the white fly, because the early detection, like a cancer, early detection is the one of the way to control the viruses. Once the virus is entered in the field, it will be appeared in the margin of the field, in the outside the field. Okay, in the margin of the field. Slowly it will be entered to the entire area. So within 5 days to 10 days, the entire area, means that is a 1 acre or 2 acre of chili plantation or the tomato plantation, it is entirely covered. So in the here we are, what we are suggesting is the early diagnosis. Once if you see the viruses based on the symptom, immediately you can pluck it and it will go for the dry it, and it will be burn it. That is a very important criteria. It won't be burned, it will be debris will be there, because already I told the virus is survive in many years. Means it will be survive in the debris of the plant. So once in the infected plants, if you identify, you can go for the burning. Otherwise, it is very difficult to control these viruses. So that is the second part is over. Now we can go for the third part. Okay, the coronaviruses. We know the coronavirus, whatever the thing is there. The last 3 months we are facing the problems. So the first was severe acute respiratory syndrome coronavirus SARS-CoV-2 in 2002. In the infected nearly 8,000 people and killed nearly 800 people. So this was followed by the 2012. That is the Middle East respiratory syndrome, MERS, COVID in 2012. It had, but it had difficult to transmit it by it is a highly lethal virus with nearly 2,294 cases in 2019, so and 35% mortality. The third of the viruses now we are facing that is the SARS-CoV-2 is the severe respiratory disease COVID-19. So it was first reported in China in late December 2009. So this virus is the enveloped virus. Already I told the classification. Then positive sense oriented single strand RNA viruses. Here also the single strand. Here it is the RNA virus, but in the plant that is the DNA virus. Here it is a single strand DNA virus, subfamily of the coronavirus. Four genera of COVID coronavirus: alpha coronavirus, beta coronavirus, delta coronavirus, and gamma coronavirus. Four type is available. That is the coronavirus. This group of virus is the zoonotic origins from the animal to human being. Alpha, beta, gamma, and delta. So that is the thing, Sam. So the structure of the virus is the SARS coronabacteria virus, which is discovered in the Wuhan city jungle. So that the positive sense single strand RNA. The size of the virus is 80 to 160 nanometer length. So that is a very important 80 to 160 nanometer length. The genome is around 29,891 nucleotides. That the plant DNA is, I told, that is a 3,000 base pair. Here is a 10 times higher. That is the biggest viruses. We are considering that our aspect that SARS-CoV-2 is a single strand positive sense RNA, 30,000 kilobytes, 30,000 base per viruses. Maybe very big size of the viruses. But these viruses contain four structural proteins. That is the envelope proteins, that is called E. In plant virus also, we differentiate three or more proteins. No? Like that, here also, the envelope protein, spike protein, membrane protein, and nucleocapsid proteins. These are the four important proteins. Other than non-structural protein also is there. But the M protein is the most abundant, mostly responsible for shape and envelope. The E protein, that is the envelope protein, is smallest structural protein. So our concern is the spike protein is the one of the important criteria for this COVID-2. So the spike and movement protein also is the transmembrane proteins. So is membrane proteins. Yeah, which one? That is the yes, membrane and spike proteins is the proteins are also the transmembrane protein that involved in virus assembly during replications. So the nuclear proteins remain associated with the RNA forming nucleocapsids, and although also the nuclear capsid proteins also is involved for the replication cycle and host cellular response to viral infections. So the polymers of S protein remains embedded enveloping the crown-like appearance. That is why it is called the coronavirus. The crown-like appearance is appeared in the surface of the proteins, surface of the viruses. So that is why it is called the coronavirus. So that is called the S protein. That is the things. Okay, spike proteins. So these are the lineage of the structure. And then so that is the 30,000 base per, around 30,000 base per. That is the 29,674 base pairs. The ORF1 and ORF2, these are the two protein is the major peptide is occupied around 21,000 base pairs. The remaining 8,000 base pair or 7,000 base pair is the other protein. Particularly the spike protein is the very important protein. This spike protein is the just 3,000 or 4,000 base pairs, from 21,563 base pair to 25,384 base pairs. So these are the spike protein is very important because the spike protein is entered the first process of the attachment of the bacteria virus as host. So this spike protein is attached to the host cells. So that is why it is very important. So these are the spike glycoproteins. These like a spike glycoprotein attached to the receptor of the host cells. Okay, so after that only the other protein is important. So the initial important is the spike glycoproteins. So this is the surface proteins uh, of this ones. So this is the coronavirus protein visualizations. uh, so the spike glycoprotein is comprised of the S1 and S2 in its two subunits there. So S1 subunit contains the single peptides followed by the N-terminal receptor binding domain. So the receptor binding domain is appeared in this S1 subunit. But in case of the S2 subunit, the fusion peptide transmembrane domain is there, and it is cytoplasmic domain also is there. So the first initial receptor binding domain is a very important to bind the domain. So the S1 subunit is the very important, and the S2 subunit after the binding the domain, the it is transferred to the cytoplasm. So that is the important for this one. So the S2 subunit highly conserved region, 90% antigens with the bats also, SARS like COVID and human SARS-CoV. So the S2 subunit is common for the things. So once the spike glycoprotein binds to the ACE2 receptor, so this ACE2 receptor is present in the human cells. So here the researchers is nowadays the researchers are working in this area. How the interaction between the how the proteins. Sorry, disturbance is there. So now the researchers are working on this how the glycoproteins now how the glycoproteins is bind to the human ACE2 receptors. So that is why this is working, and if you break this here, it is very easy to control these viruses. But it is very difficult because in the bacteria or fungus, to eliminate is possible, very easily possible, because if you cleave the surface or if you lyse the surface of the cell wall, definitely we will kill the bacteria and the fungus. But here it is nothing is there. There is no cell wall. There is no cell is there. Cell wall is there. Only the replication. If you eliminate the viruses, you can eliminate this receptor, or otherwise if you inhibit the replication mechanism. But it is very difficult also. Inhibit the replication mechanism also, it is very difficult issues. That is why the most of the researchers worldwide they are working in these regions, because the ACE2, that is the receptor present in the all the types of all many cell types, particularly lungs, heart, blood vessels, kidney, liver, and gastrointestinal tract. So you can imagine. So the receptor is appeared in the is available in the all the regions. So means the virus is easily attracted in the all the regions, either lung or heart or blood vessel or kidney or liver or gastrointestinal is easily bind with the viruses. So it is present in the epithelial cell, which line certain issues and creative protective barriers. So the major function of the ACE2, that is the thing is exchange of oxygen and carbon dioxide between the lungs and the blood vessels. That is the things appeared. Okay, exchange of the oxygen and the carbon dioxide. In the lungs, ACE2 is highly abundant. That is why this SARS syndrome, COVID is infected in the lungs, because it is highly abundant in the things and on type of two phenomena. So oxygen, where oxygen is absorbed and waste carbon dioxide is released by the function of the ACE2. So the ACE2 major biochemical pathway is regulating the process of the some vital element. So ACE2 is the vital element, is the biochemical pathway that is the critical regulating process. Regulating the angiotensin. So regulating the blood pressure, wound healing, inflammation, and renin-angiotensin arterial systems pathway. So the ACE2 is not properly function, it won't be regulated this blood pressure and wound healing. So the ACE2 helps modulate the many activities of a protein called angiotensin, This angiotensin 2, particularly 2, that responsible and it is increase the blood pressure and the inflammation and increasing damage to the blood vessels and various types of tissue injury. To control the angiotensin, ACE2 is the very important factor. So what we have doing, what is the ACE2 is thing, what is the function of the ACE2 is, the ACE2 convert the ANG, that is the angiotensin ANG, to other molecule to counteract the effect of ANG. It won't be counteract, it will be automatically the blood pressure and other thing it will be increased. So the ACE2 major function is convert the ANG. Okay, so after greatest variant COVID-19, ANG can increase the inflammation and the death of the cells in alveoli, particular area, packet, which are critical for bringing oxygen into the body. So the harmful effect of ANG and reduce the ACE2. Harmful effect reduced by ACE2. So when the SARS-CoV-2 virus bind to the ACE2, so it prevents the ACE2 from performing normal function to regulate the ANG signaling. So once the SARS-CoV-2 bind the ACE2, it is prevented, it is inhibited. The ACE2 action also is inhibited by the course COVID-19, the COVID-2. So removing the brake from the ANG signaling and making more ANG available to the injury tissues. So this is the major causes. So this ACE2 function is inhibited. ACE2 function is inhibited by the ANG. That is why you will get the more diseases and more things occur. So these are the overall picture of how the coronavirus is entered to the thing receptors. Once the receptors, ACE2 receptors is attached to the spike proteins. Okay, this is a spike protein. That is why it is called the coronavirus, crown-like structure. So it is attached and it will be moved to the tissue, and once it enter, the release of the viral genome, the single-stranded viral genome positive sense. So after the viral strand, the single-stranded genome RNA is translated and it will be form a negative strand also. Once this is a negative strand is formed, replication is started, then it will go for the subgenomic RNA. So subgenomic RNA, the genome replication is occurred. This genome replication is dependent on the RNA dependent RNA polymerase in the host mechanism. Then the translation of over, the transcription is over, then it will go for the translation. The protein is formed in the endoplasmic reticulum. Then once the from the endoplasmic reticulum will go for the endoplasmic mitochondria, then it will be formation of mature viron. So once the formation of mature viron, it will be destroy the cells and it will move to the exocytosis and move from one tissue to another tissue. So this is the mechanism behind the COVID. So the major problem is the spike proteins. The spike protein is around 3,000 base per spike proteins. So before the China, it will be the strain is vary from the Europe strain. Is now we are facing the Europe strain. So that is the different type of strain is involved in the India also. So it is that is why it is the thing is possible. So the translation of subgenomic. So that is the picture of the things. Okay. So this is the viruses. So the viruses is the very complicated genomes. So because the different genome organization, either it's a single strand to double strand RNA, single strand RNA, that is why the people are not ready to work because even the plant virus also, so because the plant virus is not causing to the human being, is not transmitting to the human being, even though the complications because why we are choosing the DNA virus means that is That is we can go easily isolate the DNA. In case if you go for the RNA virus, it is little bit difficult. So the major problem is the little bit difficult. Even though if you the DNA virus, the isolation and the identification is easy, but the reconfirmation is very difficult because we need the vector for the DNA viruses. Okay, because the DNA virus transmitted by the vector. So the confirmation part, isolation and identification part in the plant viruses is little bit easy, but for the transmission part, confirmation part is very hard because we need a white fly. So the plant, if you virologist is working, they have to understand the animal also, white fly also, and the plant also. But same time in the animal viruses, particularly these human viruses, it is very difficult to manage. So such a kind of system is not available. Only the King Institute is in Guinea. King Institute is available. The entire full sophisticated laboratory facility is there. So there is no facilities available in the Madras University or other universities to handle the animal viruses. So such a kind of problem is there in the virus handling process. So I think my part is over. I hope you are all understand this basic aspect of the viruses. Why I am introduced the plant virus, I mean that is my area. Last 10 years we are working. Nearly 2 PhDs to 3 PhD students is completed this area. And another one area we are working on the nanotechnology, because nanotechnology is interrelated to the plant viruses. That is our area. We are not working the drug delivery system or clinical aspect. We are working focus only in the nanotechnology in viruses, because the identification of viruses, usually we are going for the PCR and other things. But to avoiding the PCR and other things, we are using the nanotechnology. So nanomaterial encapsulated with the DNA, that kind of work, nano-capsulated antibody, that kind of work we are our laboratory is going on. If we any help or anything, if you want to work on this kind of area, please approach me. So once again, I thank the organizers to share my views and other things. Thank you for your patience.
Thank you.
Any clarifications? Please make it. Please, ma'am.
Yes, sir. Thank you, sir. Thank you for your speech. It was very interesting and informative. We learned a lot of viruses from your speech, sir. Thank you, sir. Thank you.
Thank you, ma'am. Thank you. Thank you, sir.
We have come to our end of our program. It's time to propose vote of thanks. Now I invite Dr. B. Kalaivani, Assistant Professor of Zoology, to propose vote of thanks.
Good evening to one and all. Saying thank you is more than good manner, it is spirituality. I thank our management for permitting us to conduct this webinar and their support in all our activities. I am very much grateful to our Madam Principal, Dr. R. Umarani, whose guidance, support and encouragement. I also thank our Controller of Examinations, Dr. E. Roja, for her support. I am very much thankful to faculty members from our department, other departments for their support. I am happy to thank Dr. L. Akilandeshwari, Assistant Professor of Chemistry, to provide this Zoom platform. Last but not the least, I thank all the participants who attended this international webinar and made the program a grand success. Thank you.
Thank you, participants. யாரு அவளே எடுத்துட்டாளா?
என்னது?
Dear participants, through certification, we can get 100 certificates daily. So fill up your feedback. Definitely you will get your certificates within 5 to 6 days. Thank you.
அனுப்பல அவங்க 25 பேர் இருக்காங்க 100 பேர் இருந்தாங்கன்னா போதும்